All right, so we are now on I think this is the correct one. I think we're on circuitry 2. We're going to go to the bipolar cells because we did generalized circuitry before. And I think that, it's always good to go through now each of the individual cells as best as, I know them, so that it makes sense in a framework when you start thinking about the retina. So let's move. So I said this to you before, and I'll say it again. We are trying to basically show you the same thing over and over again, and then teach you a lot more than that. So then when you get back to the thing that you were supposed to know, it makes it a little bit easier because it seems pretty simple. So we're trying to go and make sure that you know these layers. And so we're going to teach you about everything in those layers. So again, as I showed you before, that's what we're looking at. And then, when we construct the retina, I just want you to remember that the retinal circuitry is complicated, even though I showed you that there were 7 cells and the sort of generic cells. It's complicated. So, don't ever feel like you don't understand it and everyone else does, because it actually isn't completely understood. And every day there's like a new, or maybe not every day, but maybe every year there's something new that's discovered about it. It's not, like some of the other organs where we kind of know a lot about it. The retina is it's still a mystery. So what I always say is you try to break it up into individual pieces, and then we just keep repeating this from a different angle. So in that vein, I think we remember, and we're going to review if we don't, sort of what happens to all of the things that I describe in terms of activation or deactivation in terms of the individual cells like we did last time. So remember, this is dark. So in a light state, and remember we talked about there's a difference between the central photoreceptor and the surrounding photoreceptors, and then all the cells that interact with them. So tell me, in the light, what is the status of the central photoreceptor? Is it active or inactive?
It's deactivated in light.
Correct. It's deactivated in light, right? And that means that the bipolar on cell is active or not active.
The bipolar on is active.
Correct. So bipolar on cell is active. And then what happens to the horizontal cells that are kind of communicating with the photoreceptor? If the central photoreceptor is inactive, what is going on with the horizontal cells? Are they active or inactive, the ones that connect to it?
So they are the opposite. So they are active to try and pick up the surrounding signal.
Right. So remember, the horizontal cell is like the guy at your house when you have a party. If you're the central photoreceptor, he agrees with you. He has the same kind of valence that you do, but then he goes and flips the guys around. So you remember that? So in this case, the horizontal cell is actually inactive because the central photoreceptor is inactive, but then he turns around and activates the surrounding photoreceptors. Do you remember this?
Yes. Yes.
Right.
Yes.
So it's that lateral inhibition, or in this case, lateral activation, if you want to call it, where the central photoreceptor is shown to be whatever its valence or its polarization state is. It's even more obvious because the surrounding ones are the opposite, and the horizontal cell is going to help with that by basically inverting the surrounding photoreceptors, because it is taking the same polarity as the central photoreceptor because they're both at the same party, so to speak. That makes sense?
Yep. Yep.
Okay. And then the bipolar cell then, which is on, which one of the ganglion cells is that bipolar cell going to activate? On or off?
The bipolar on is active in the light state.
Correct. And then which one of the ganglion cells then does it activate? The ganglion cell on or the ganglion cell off? Do you remember that? So
ganglion cell, it's the same.
It's the same.
Yes, exactly. It's the ganglion cell on cell.
Correct. So the ganglion cell on is going to be active and the ganglion cell off is going to be inactive. And then, of course, you have these amacrine cells that these guys interface with. We don't really know the polarity of those because they're very different. And then, of course, they help out over here. And then, of course, whatever signal then from the ganglion cell on or the on cell will be sent to the LGN, okay? This is kind of like the way the schema from a structural point of view. And then if we do this and we basically we figure out where their nuclei are. So we take each line here, and we say, okay, well, that's pretty much the outer nuclear layer. If we talk about where they live, remember the photoreceptors live in the garden kind of apartment, the outer nuclear layer, and then the outer plexiform layer, which is a bunch of axons, is where all these connections are happening, right? Because they're the connections between the photoreceptors, the bipolar cells, and the horizontal cells. And then, of course, if we drop down the amacrine cells, sort of their nuclei into this zone, this is where like all of the masses live in the inner nuclear layer. And then, of course, the inner plexiform layers where the amacrine cells, the bipolar cells, and the ganglion cells all interface. The ganglion cells live in the penthouse apartment of the ganglion cell layer. And then, of course, the nerve fiber layer is what sends it to the LGN. Does this schematic make sense?
Yeah.
Right. So it's just a different way of looking at it. And I think it's very useful to do it like this. And so we'll keep doing it like this. So today, and then let's go through the construction of what I did schematically here. Let's just build it again. That's your photoreceptor. That's your bipolar cell. That's your horizontal cell. That's your ganglion cell. That's your amacrine cell. That's your Mueller cell. That's your RPE. And then there you go. So, we just named them all and it makes sense now structurally. Okay, so that's very helpful. So now we're not going to go through photoreceptors because I do that when we talk about phototransduction. The photoreceptor is complicated, no doubt, but it's mainly complicated in its receiving of light action, and less so the secretion sort of once that impulse is made, the secretion that it does of the neurotransmitter to its distal kind of interneurons. Just remind me again, what is the neurotransmitter that photoreceptors secrete?
Glutamate.
Good, glutamate. That does not change. It's not because today's a Monday, now it's different, right? It is glutamate. That's the answer, and then when it secretes it and when it does it based on the light. Okay, but what we're going to focus on is the next guy in the chain, the bipolar cells. So let's talk about the bipolar cells in depth. So they're an interneuron, as I told you, which means one neuron to another neuron. There are 36 million cells that we said in general. Remember, the photoreceptors are split into rods and cones, and there are 120 million rods, 6 million cones. So 126 million rods and cones are sending all their information to these bipolar cells, of which there are only, quote unquote, only 36 million of them. Okay. Transmits from photoreceptors to ganglion cells. Now, when I said there were greater than 10 total, I just meant the types. So, of each of the 36 million cells are going to fall into a type here, and we'll see that. Now, that there are on and off forms. That's a different kind of way of describing them, and we'll go into why. We know that glutamate inhibits the on forms. Why is that true? Because glutamate is secreted when the photoreceptor is in a dark state. And so thus that secretion of glutamate is not going to activate the on form because light, quote unquote, is not on at that point. So it activates the off form. And so we go through this again from another angle. In the dark state, glutamate is secreted. If glutamate's being secreted, which of the bipolar cells is activated, on or off? Let me know.
Bipolar off cell.
Got it. Off cell is activated in the light state. Is glutamate secreted or not secreted by the photoreceptor?
Not secreted.
Correct, not secreted. And then which of the bipolar cells is activated, on or off?
The on cell.
Correct, the on cell. So we have the on cell activated. But we're going to go into this more in depth. And I flipped this up here. And I just want to say, I'm going to knock out some of these things, just reread some of the new parts here. It doesn't really transition well. But for the most part, we say that there are 36 million cells. And, they're an interneuron, all this stuff. But there are some exceptions to how many classes there are. And I'm just going to tell you that there are actually 11 total classes, 10 synapses with cones. Mainly I say 10 because the ones where there's heterogeneity is whenever they interface with cones. But there's just one rod specific, a very weird rod specific bipolar cell. And then I just wanted to mention, again, I say transmits from one neuron to another, but there are some exceptions to that, very rare ones. So let's go into some of these, okay? Now, we know there's on versus off, and I just literally am hammering that into your head because it's important, because in some future date, you're going to understand what that means. But there's also other ways of describing it. So there's a diffuse bipolar cell and there's a midget bipolar cell. There's a flat bipolar cell and there's an invaginating bipolar cell. And so you can get this kind of this you can mix and match in some settings. So it's a flat midget on, and then I use the word center. So that's actually what on means, on center. I'm just letting you know, I'm sort of building for you the nomenclature, but it's flat midget on center bipolar cell. That's a thing. Okay. Like you're not, it's not that like you're, I don't know, you're flat, but you can't be off or something. I mean, all I'm saying is that those are just like mix and match ingredients. Now they all don't exist, but that's what I mean. They're different characteristics of a bipolar cell. The vast majority, to belabor the point, the vast majority of the types of bipolar cells synapse with cones. This is a direct relation to the fact that cones really are what we use to see fine vision. And so you're going to want a lot of their information being transmitted up the chain. Not that rods are not important. They're incredibly important. But cones are where we really are importantly are able to use our central vision. And that's why it's very important that most of the bipolar cell apparatus is kind of dedicated to it. But in terms of the types, but remember, there are 10 classes that are basically for cones, and then one class for rods that are rod specific. So let's go and think about what on versus off is more in depth. Conceptually, you can keep thinking about it as on and off to light, the way everyone inherently and sort of intuitively thinks about it. But just remember what it means is on center and off center. So I'm going to give you a schematic here. Now that blue, navy blue triangle, that's a cone. Okay, it makes sense because it looks like a cone. Okay, so that's a photoreceptor, if you can imagine. And then you got these blue squares on and off, which are going to represent your on and off bipolar cells, as I called it. But now we realize what it's called. It's on center and off center. So in order to kind of understand this, we already have a sense of the surrounding photoreceptors. So let's put those guys in there kind of as a representative. So you have these two surrounding photoreceptors, and then you have each one of them, let's say with a single on and a single off. Let's just do it so it's easy, of a bipolar cell connection. And then I send light to the center guy. Okay. So light right now, in this case, is shining on this middle photoreceptor. So he goes, the light shines down on this middle photoreceptor. So then if we look, which of the bipolar cells are going to be active in this state? Well, this is what's going to happen. You're going to get the on-center bipolar cell activated for the set when we look at the central photoreceptor, but then we look at the surrounding or the off-center photoreceptors, and the off bipolar cells are active. Okay, but then let's this makes sense, but let's like try to confuse ourselves, even though I don't think it's going to be confusing. I'm going to redo this, but then say it's going to actually go over here. So now the light's over here. Okay, we just like had two flashes of light. The first flash was in the center, but now the second flash is going to be quote unquote on the side, quote unquote not on the center. So we have this, and then we say, okay, what's going on with terms of the bipolar activation? And so we say, okay, well, this is what it'll look like if you were to check which of the bipolar cells are active and which of the bipolar cells are inactive. Does that make sense?
Yeah, absolutely. Because of the horizontal cells.
The horizontal cells play into this. You're exactly right, 100%. But remember, that's exactly right. You got it. So the horizontal cells are also playing into this. But then just remember, at a native state, right? The surrounding photoreceptors are also going to be, in this case, secreting glutamate, right? Because they don't have light being shined on them. So they are going to be active no matter what, which will then activate the bipolar off cell, while the central photoreceptor, in this case on the side, is going to be quote unquote inactivated, which means glutamate stops, which means the on bipolar cell continues to be activated. So you don't even need a horizontal cell here to potentiate to do it, but it potentiates it. Does that make sense?
Yep. We're assuming that the middle and far right photoreceptors are in a dark state at this point.
Correct. So, exactly, because they're off center to the light. If you created this situation where it kind of sensed that there was light off to the side. I'm trying to introduce this concept of like the photoreceptor and the apparatus. It's not just dark or light. It's like, oh, I'm not only dark, I'm also dark, and the guy over there is is lit up. I have the capacity to know that through some mechanism. Does that make sense?
Mm-hmm.
Good. So there you go. So that's how we do it. So I'm showing you this. And one of the things, it's like, okay, well, Dr. D, you're telling me that, if I didn't know, the only way I could tell whether a bipolar cell, just by looking at it, is on or off is by shining a light and then telling me which one's active or inactive based on where you shine the light, like I just showed you. You're like, okay, well, that experiment I could do, But, it would be nice if there was a way that I didn't have to shine light, maybe an anatomic way, right? To know whether something's on or off. That would be pretty cool, if it was that way. Otherwise, we'd always have to shine light to see which one was on and which one was off. I'd see two bipolar cells and they don't have signs on them that say off and on. It's just like two bipolar cells and I have to physiologically determine it. And I would love to have another way to do it so I don't have to physiologically do that. Thus, I could use pathology to figure it out, right? Because otherwise, I'd need it always to be in this active physiologic state. Well, the question is there a way to know this? And obviously, the answer is yes. Otherwise, I wouldn't have asked this question. So what exactly do these bipolar cells look like? Because I've drawn them like this, but is that actually how they're drawn? And so the answer actually is, if we put that cone there again, is that they're not actually drawn like this. We're actually going to draw here and say, okay, well, where is the cone as I drew for you there in the outer nuclear layer? Assume that's where the nucleus is. And then where the interface, basically the axon connections between photoreceptor and bipolar cell, where is that? Well, that's in the outer plexiform layer because we know that's axons. And then, of course, we know all these guys live in the inner nuclear layer. But then let me draw the inner plexiform layer a little larger. So just so it's more to scale, because we're going to go somewhere with this. And then I'm going to just draw a line in between, kind of randomly in the center, just, take it from here. And in reality, and I'll name this part sublamina A and sublamina B. You don't have to know why I do, just like section A and section B, we call them sublaminas because it's a laminar kind of plane here. But let's say sublamina A, sublamina B. And then I say to myself, okay, well, this is what the on cell tends to look like. So then, you say to yourself, oh, okay, this is how I'm supposed to, I could take a pathology specimen and lay out amount of the retina. And then if I could figure out where was sublamina B and sublamina A, I could say, okay, well, the bipolar cell that is kind of has its distal end or its axonal connection with whoever he's going to connect with in the inner plexiform layer. If that's in sublamina B, he's an on. And then the other guy is an off. Does this make sense?
Yeah.
Okay. So on center cells, just to show you, and I say it in a different way, have their ganglion cell synapses, because again, who are they synapsing with? Well, okay, they're amacrine cells, but the main chain is photoreceptor to bipolar cell to ganglion cell. So where is the on center ganglion cell going to synapse? In sublamina B, which is the distal IPL, or the one that's closest to the ganglion cell layer, if that makes sense. I think it does. And who are they synapsing with? The ganglion cells. So the ganglion cell is actually synapsing with the on-center cells in sublamina B, and then the off-center cells have their ganglion cell synapses in sublamina A, or what I call proximal IPL, meaning the one that's closer to where light is originating, okay? And just remember, that's the direction of light. So here's your cone, here's your bipolar cell, that's A and that's B. Note one, and then your ganglion cell. Notice how the A and B nomenclature follows the direction of the signal. Does that make sense? It goes from A to B as if you're starting from the cone where the photon is delivering its electrical signal and shooting it up towards the inner retina where it's going to go to the brain. And you can see A and B are listed in that order. Does that make sense? So whenever you see the directionality of things, just remember how things are, named and numbered, and you'll understand, okay, I remember that's A and that's B. Does this make sense?
Yeah. Is there a physiological reason why the bipolar off cell synapses in sublamina A versus, yeah, and that's just the way it is. And we're just lucky that we can, they're different. So we can,
Exactly. Why do we have two kidneys? And you know what I mean? And one is slightly higher than the other, right? But yeah, this is just the way it is. And thankfully there's a method to the madness, or an elegance to it, if you will. But yeah, there's no reason as to why I would maybe understand why evolutionarily, I'm sure there's someone that's going to come up with it, we would have the off in sublamina A and the on in sublamina B. So just remember, this is just determining on and off bipolar cells spatially. I didn't say a thing about midget versus diffuse or invaginate versus flat. Those are like different qualities, It's like saying, we all live in New York City, but it doesn't mean that you can adjudicate our height on the basis of that. That's a different quality of human beings, despite the fact that we could identify our location, right? And say, okay, well, they're in New York City. Well, guess what? You can determine whether someone's on or off based on their spatial location. It doesn't tell you whether they're midget or diffuse or invaginating versus flat. So here is a schematic of the primate mammalian retina, where it's not human, okay? So it's not exactly, but this is the primate retina, which is close enough to being human. And you'll note there are not, I mean, I'm listing for you all of them. I mean, these are not exactly all of them, but there's not that many other types. Here you go, right? So notice something. Okay, so there's 11 different ones, as I told you, and look at where they're separated spatially in the IPL. Now, one thing I need you to understand is this photo is upside down. It always is drawn this way because us as retina specialists and ophthalmologists, for whatever reason, we gave up the conventional naming strategy to the neuroscientists. And the neuroscientists always start with the photoreceptor on top, whereas we start with the photoreceptor on the bottom because it's ergonomic. That's the way you do surgery. It looks like it's on the bottom of the glass when you do surgery on the retina, but it's not us that defines convention. So notice what's going on here. The off layer is actually at the top and the on layer is at the bottom. Does that, because it's reversed. The ganglion cells at the bottom. You always have to know this when you're reading any of the literature if you ever choose to do so. The vision scientists invert it. I happen to believe they're wrong because we should be defining it because we do the surgery, okay? But I don't make the rules. So you just have to know which way it's going. Now, notice something. DB, in this case, the acronyms show diffuse bipolar. So you have this diffuse bipolar. Whatever diffuse means, it's a class, but notice how it can both be, in this case, it's off, and then it can also be on. And how do you know that? You just can look at where its axons are. Look at the final the protruding axon. I can tell you without you telling me off or on layer anymore, because I know that if you're in the proximal IPL, meaning the one that's closer to the bipolar cells or closer to the inner nuclear layer where the nuclei of the bipolar cells reside, I know for a fact that is going to be a bipolar off cell. And then I know for a fact that if it's protruding all the way through to the distal IPL or closer to the ganglion cell layer, then I know that this one's going to be a bipolar on cell. Doesn't matter if they're diffuse or not. Does this make sense?
Yeah.
Good. So we're going to now talk about the other qualities because those are important. So you have flat versus invaginating bipolar cells, okay? And what does that mean? Flat just means on the surface of a cone. Makes complete sense because it's flat. Invaginating means within the cone to the synaptic ribbon, whatever that means. Just know that that's what it is, okay? The cone to the synaptic ribbon. Here is a picture from your book that you've seen before that I showed you, and you may have read about it, but now we can make sense of it a little bit, right? So if we say to ourselves, what does FB stand for? It stands for flat bipolar cell, but what does IMB stand for? It stands for invaginating, and then the M we'll talk about, but bipolar cell. So FB is flat bipolar cell. IB in this case is invaginating bipolar cell. Now, you know, they're all bipolar cells, but they have that one different little characteristic that, again, you can see now in this case, you know how it's being defined as spatially. You can see it in pathology. Does this make sense?
Yes. Yes.
Okay. So now you know that. Okay, so we did flat versus invaginating. Now let's talk about midget versus diffuse. So a midget, just this is the way they're defined, is specialized to only one cone, whereas diffuse means multiple photoreceptors, usually cones. So here is your your situation here. You see FMB, flat midget bipolar cell, right? Flat midget bipolar cell. And that just shows you that you can have both types. And of course, that IMB stands for invaginating midget bipolar cell. Make sense?
Yes.
Just the fact that that exists, not why. I'm just telling you that that exists.
Yeah, absolutely. Absolutely. Yeah, this makes sense.
So now you can see it and you say, okay, I know what those mean. Now I'm not entirely sure why it matters, but I know it means that. Okay. Okay. Well, diffuse is usually multiple. So here, let's look at a diffuse. Let's look at kind of a different way of looking at these photoreceptors and the retina. Note again that this is upside down because it wasn't done by MDs. It was done by PhDs. So that's how you can tell. And so here's your fovea up top. And then you can see IMB in this case is invaginating midget bipolar cell. So I'm going to go and like circle around here, invaginating midget bipolar cell. But what did we, and then we go over here and we see FMB be flat midget bipolar cell, right? But remember, midget bipolar cells synapse only with one cone. I just told you that. So just think about it as like a midget. It's small. That's why they define it as midget, because it's a small cell in terms of size, but it's only one cone. And remember, that's if you want to remember how a neuron works, and all of these are neurons, the bipolar cell is an interneuron, but all neurons have a dendrite, which is where it starts, and then where it gets its information, and then it has an axon. I mean, like that goes, kind of forward, and that's where it's delivering itself. So we're really talking about the bipolar cell's connection to the photoreceptors. So what are we talking about? The dendrites. So the dendritic connection of the midget bipolar cell is only going to connect to one cone. And so if every time you see the MB, that's the only connection that B has, right? Because it's a midget bipolar cell and it's connected to that cone. Does that make sense?
Yes.
Okay. So if we just count it out, anytime I go and I show you, I go, okay, well, here's an IMB1 and IMB2. Well, if I follow it back, there sure as heck better not be a split here where it's like connecting to something else. I don't want to see that. Because that would be a problem. That would invalidate the midget status of this bipolar cell. And so, that's what these guys do. Okay. But then note the flip. Okay. Just the other side of it is not true. One cone can synapse with multiple midget bipolar cells, right? So one cone, multiple midget bipolar cells. So, I use this, I don't know if this is uncouth here, but I say, midgets are monogamous. We have only one cone, but cones are creeps. They can be with multiple midgets, if you will. So if you look here's, let's say, a cone, and notice how this one cone has the dendritic processes of one midget and a second midget. Does that make sense?
Yes.
Okay. So this is just showing you that these bipolar cells are being specialized to just one cone in this case. So this cone must be super special. And then these midget bipolar cells really go and then do whatever it is that they do elsewhere, right? But my broader point is that's just showing you how some photoreceptors are privileged over others in terms of information gathering, in particular cones, which makes sense because that's central vision. So there you get the structure and then the function. Okay. So now let's look at a weird example. And the only reason why it would be much easier if I had a midget bipolar on cell example of physiology, but I don't. So, it's targeting one cone in this case. So they did this I'm showing you these classic experiments from the 50s, 60s, and 70s. A lot of the people who did this, our field is filled with these people who won Nobel Prizes multiple times because they did these very classic experiments on different animals and, and then determined how the brain worked basically and neurons worked because they used vision as their apparatus to do so. So here's one of these. This is confusing, so just work with me. You're gonna have to kind of get in your head what off center means and on center. It'd be much easier if they tested this with on center, but of course that would be kind of obvious. So I guess they showed the off center kind of physiology. So if we look and we're targeting one cone, here is a shot. This is the light that's being shown, like how the light looks. So in this case, a spot center shined light shows that the center spot is lit up and then the surrounding annulus is not lit up. So the off-center kind of cells are bathed in darkness in this case. Okay, so got it? Okay, so if we do this, and then this is the opposite, where the annulus is lit up. So all of those photoreceptors that are in the annulus are getting lit up, but then it's still referring to the cells in the center. So we're targeting right now, we're measuring a midget bipolar cell that is off center, that is connected to the center photoreceptor in both cases. Does this make sense? I'm just going to explain to you what this graph shows, but do you understand what I'm what example, which cell this is putting its probe into? So
yeah, we're just focusing still on the center photoreceptor cell, whether it's bathed in light versus dark.
Right, and it's not the photoreceptor per se, but it's the midget bipolar off cell that is connected to the central photoreceptor, which in this case is one. Okay. Does that make sense?
Yes.
Okay, so what we're doing then is we're going to do that this bottom binary kind of signal type, on-off graph is showing you what the what is happening in terms of the light in the center here. Okay, so it's showing you light in the center. It's illuminating, and then now it's not in the center, and then it's illuminating, and then now it's not in the center. Does that make sense? That's what this is just showing it. And they're turning that spotlight on, and then spotlight off, and then spotlight on, and then spotlight off. Okay, that's what they're showing here. And then this is actually measuring the polarization or the activation, okay, of the midget bipolar off cell. Okay.
Got it.
So if I turn on, I'm going to use the word on because it's a little easier in this case, but if I turn on the light in the spot center, then you get what's called hyperpolarization. Remember, depolarization is up here because that's more positive. If this was a y-axis, down here would be minus, and then up here would be going in the positive direction. So if I depolarize, I go up. If I hyperpolarize or I become inactive, I go down, more negative. Does that make sense? Which direction does that? You remember that, right?
Yep.
So when it goes down here, this means inactive. When it goes up here, it means active, because this means depolarized. This means hyperpolarized. Okay, so when spotlight is on in the center, on meaning it's illuminated, my guy here, this off midget bipolar cell, is inactive, and that makes sense, correct?
Yes.
Okay, but then I shut off, like I turn off the spot, and then relatively speaking, now he's happier, so he becomes depolarized. Does that kind of make sense?
Yep.
Okay, so in this situation, the spot is going on and off on its own, and it's just to show you just how sophisticated the midget bipolar these cells are. So in this case, I'm turning the spot on and then the spot off, and then the spot on and then the spot off. Notice I'm not lighting up the annulus ever. I'm just doing on and off of the spot, and notice he is responding to that delta in the correct fashion. When the spot center is on, he's not happy, so to speak. He's inactive. When the spot center goes off, well, now he's relatively way more happy, and he'll show you that occurred. Hey, I'm going to show you that happened. But then let's go to the annulus. And I know I'm belaboring the point here, but it's just showing how sophisticated this is.
Yeah, I'm wondering what the difference would be, because you kind of get on off in example A, but the difference is the annulus, I guess.
Right, but I was going to say, notice how the waveform is not different in terms of just the polarization. The polarization kind of matches, but it's inverted because it does respond to the annulus. You turn on the annulus, he becomes depolarized, right? Because he's the center off. So he sees off center is happening. Does that make sense?
Oh, in example B, we're toggling the annulus.
You got it.
Always dark. Got it. Yeah.
Got it. But he's still in darkness here. He'd never change it. Like the light that's being shined on him does not change, because he is a center cone midget bipolar off cell. So he's sitting there still connected. No light is bathing or unbathing him. But what is he sensing? He's sensing the annulus going on and off. And if you turn on the annulus, he's happier because he's an off center bipolar cell. So he likes when the off center is illuminated. And so he depolarizes, and then you turn off the annulus and he hyperpolarizes. Does that make sense?
Yeah.
So notice how his responses are inverted here, but he shows a response on either situation because he can sense both the spot delta and the annulus delta.
That's tricky. Yeah.
Isn't that cool? And so from my perspective, and by the way, this would be completely inverted for the midget bipolar on cell. By the way, it's just the bipolar on, bipolar off. The reason why they picked the midget bipolar cell is because they know it only has one cone, so they could toggle one cone.
Yeah.
Makes sense? So that makes sense why they did all this. So I just like to do this because now it's like, yeah, I get it. Like if I wanted to toggle something and I wanted to stratify, I don't want to use a diffuse bipolar cell because he could be getting information from a bunch of different cones. I want to just control one cone and then see what that one bipolar cell does. Well, then I got to pick a midget bipolar cell. And in this case, I wanted to do off center. So I picked a midget bipolar off center cell. Make sense?
Yes.
Cool. Yeah, it's confusing, but I think it's starting to make it's once you get it, it's like, wow, that's just wow. I mean, like that it can do this, right?
Yeah, it's cool. In the example we had earlier in the presentation, we were changing the light between the center photoreceptor and the ones on the side, but basically whatever one. But we didn't really talk about that's distinct from talking about the annulus. Like in those.
Well, no, it actually still is consistent. So if we go back to that, and I will do this just because it makes sense. Remember, when the annulus is lit up, the center off is then turned on, meaning he is active, right? I mean, that's what we're doing. The annulus is toggled to on, and he is active, right? So just remember that. Now, if I if I go all the way back, and I'm going to do this because I think it'll be instructive. If I go all the way back here and I say to myself, okay, well, let's go ahead and let me just do this. Let's go ahead. This is okay. And I say play from current slide. And I say, okay, I'm going to do this guy. So now, and then we've got this, right? But then let's do this. And I'm going to shine. This is like shining light on the annulus.
On the annulus, yes.
But remember, we were focused on this bipolar cell.
The bipolar off cell, right, in the center.
Above the central photoreceptor.
Yes.
And so if I show you what happens, he is active.
He's active. There you go. Yep.
And so it's totally like makes sense, but it's just from different angles, right? I showed you the canonical kind of seminal paper, I think that experiment that won, I believe, a Nobel Prize on this, but I also showed you this weird cartoon graphic too, right? And it kind of does it too, right? It's like both the same. Let me go back to that thing. So we'll go all the way back to our guy here. So we were here, and so, you know, I showed you this and targeting. So we get this, okay? So I think we understand that, but let's talk about that special weird, I mean, I shouldn't say weird, it's not weird, but it's this rod bipolar cell. It's very specific. It's like in the name, rod bipolar cell. I mean, you didn't hear cone bipolar cell. You heard midget, diffuse, flat, and badging, and then here's this like rod bipolar cell. Okay, well, what is that? Well, we're going to talk about this exception because it's actually really important. So it's a little strange, and it is one big exception to the rule of the canonical pathway of photoreceptor to bipolar cell to ganglion cell. I always say that is the, you know, the big three. We go from 1 to 2 to 3. That's how you score the ball. I say that, and then I give you this exception, right? Here's a bipolar cell where something weird's going to happen. This guy, first of all, he synapses with multiple rods. So in a way, he's like a diffuse, sometimes 100 rods. So he's kind of like a diffuse bipolar cell, right? Because what does the diffuse mean? Diffuse means you're connecting to lots of photoreceptors. Midget means you're monogamous with one photoreceptor. Make sense?
Mm-hmm.
Okay, so now we have a bunch of rods. This guy, this particular bipolar cell is 100 rods, right? You can get information from 100 rods. And then you say to yourself, well, who does he connect to? But he does not synapse with ganglion on cells. And he's actually a on bipolar cell. I'm just going to tell you that. Well, actually, he doesn't have to be called an on bipolar cell. He's just sort of a bipolar cell, and why he doesn't have an on or an off designation. And you sit there and you say, wait, and he also doesn't synapse with ganglion on cells or ganglion off cells. He doesn't synapse with ganglion cells at all. And this is the rod bipolar cell. It's the guy who actually connects with rods, and how we have more rods than cones, right? By far, 20 to 1. So the guy who is in charge as a bipolar cell of connecting to most of the photoreceptors is not synapsing with a ganglion cell, nor does he have an on or off configuration. And so you're like, Dr. D, you just gave me all this framework and you're like chucking it away by telling me this most famous bipolar cell. What's going on here? Why is this? Okay, well, let's first figure out what he does. Who does he synapse with? He synapses with an amacrine cell that is incredibly famous in vision science. That amacrine cell, then it goes to a nearby cone bipolar off cell. He inhibits that and then activates this cone bipolar on. Now, you don't have to get that in this moment, but just note there's this weird like handoff that happens. Rod bipolar cell doesn't go to ganglion cell. It kind of makes a detour, grabs this amacrine cell, who again, you know that is a fair point. I did tell you that bipolar cells can connect with amacrine cells. I also told you that amacrine cells are somewhere in the vicinity of a ganglion cell. So in this zone, it makes sense that there might be an amacrine cell swimming by that's going to help you out. And so what is he's doing is he's hijacking like this other system. For what purpose? We'll get into that. But remember, this only happens, and I shouldn't say remember, this only really happens during times of low light, when the cones themselves may not be activated. So what does this mean? Okay, and by the way, if we wanted to be really specific, and I'm just throwing it, it's not called this, but you would tell me there's the synaptic ribbon. This is from your book. And you see, oh, well, look at that RB. Yeah, that's a invaginating. So if you really wanted to be really specific to Dr. D, you'd be like, well, isn't this an invaginating diffuse rod bipolar cell? I'd be like, absolutely. We don't call it that because there's only one rod bipolar cell, but you are completely right from a pathologic perspective, and that means you understand it. The other thing I just want to point out very quickly is H means horizontal cell. So just to let you know that part, okay? So H means horizontal. So you're like, now you're getting all the characters in the play, right? Like you can see, and it will make sense when you read this in your book. Okay, well, let's just go through the rod bipolar cell schematically. Okay, let's talk about it again. Here's and I'm going to call him an on. I'm going to do that. Okay, here's a on, but it's the rod bipolar cell. How do we know he's connected to that's a rod? Okay, I can't tell you how many times residents will be like, what photoreceptor is that? I'm like, I draw the cones like a triangle and the rod's like a rectangle. I thought it would be clear, but maybe it's not. So here's a rod, okay? You got a rod photoreceptor connected to this bipolar cell, okay? And then we have next door, I told you that this is relevant. Let's assume that this cone, this is a rod that's sitting right next door to the cone that it's going to kind of do this craziness to, okay? And now light shines on this photoreceptor. Remember, I shine the light, in this direction. Remember, the light passes, past all this. There's that outer segment of the rod. It takes in that, absorbs that photon, then it shoots it back up, and that's what's going on. This is not hitting the actual bipolar cell, right? Because there's no photoreception by the bipolar cell. They do not have the apparatus. Only the photoreceptor has that. Okay, so we know that. So here we go. We have light, and then here comes this amacrine cell. And so you're like, okay, because the light hit the photoreceptor became what? Hyperpolarized or inactivated. Glutamate gets stopped because rods are still photoreceptors. They're not some other cell. So they stop secreting their glutamate. Something gets activated. Well, we know it's the bipolar on cell that gets activated. In this case, the rod bipolar cell, which we're calling an on cell. Okay, so now we're there. Now he's activated. Now, who is he? He's not going to go to the ganglion cell on, because I told you that doesn't happen in this case.
Right.
He's going to turn around and talk to this guy. Okay, weird. Okay, so now we're in this amacrine cell. He's activated. What does he do? Well, he then says, hey, you are off. I'm going to inhibit you, because what is the native state of this guy when there is no light on him? When there's no light on this next nearby cone, he is actually secreting glutamate. This one is actually inhibited, and this guy's actually constitutively active, correct? In a dark state. Does that make sense?
Yep.
Okay, so then in this state, here comes light, active. Well, okay, I need to somehow transmit this information to some ganglion cell somewhere. Okay, well, here, I might as well hijack this. So I'm going to block you and send the signal through your bipolar cell on.
Oh.
Okay, now the question is, why is this happening, right? So we just do this one more time. This is a little weird, right? Here's your rod bipolar cell. I'm calling him on because it makes kind of sense. Here comes the rod. Here's this nearby cone who is not sleeping, but in a dark state. Both of them are in a dark state. You have the cone that is constitutively, I will use that word, but is activated. So therefore the bipolar off cell is active because glutamate is being secreted, and the bipolar on cell that is connected to that cone is not active because glutamate is being secreted again, which is inhibitory to it. And then you have this light beam that only shoots on the rod. And then the light beam shoots, or let me not even say on the rod. Let's just say it's a ridiculously low level of light, like one single photon. And that's where we're going with this. If it's just one single photon, okay, which cell can pick that up? Guess what? It's your rod. Your cones actually don't. They're not as sensitive, oddly enough. It's your rod that does it. Your rod's actually super, super sensitive to low light. That's why we need it, and you get nyctalopia when you lose this stuff. But anyway, so I digress. So you have this situation here. One single tiny photon of light comes in. The rod knows that it's there. The cone's still sleeping, so to speak, and just secreting glutamate. And so, boom, the rod bipolar on cell gets activated. That guy then activates an amacrine cell. The amacrine cell says, I'm going to come into, I'm active. I'm going to do my job, knock off this person, kind of distal to that photoreceptor, right? Downstream of that photoreceptor. The cone doesn't know what the heck is going on, right? The cone is not being activated here. And then he goes, okay, and then I'm going to send that signal. So I've hijacked kind of this entire apparatus. And now we're going to go to the ganglion on cell that was connected to this. That makes sense, right?
Yeah, it totally does. And it probably doesn't matter whether this is a one-to-many relationship from the rod bipolar
Got it.
cell to the cones, because there's so many more rods than cones,
Got
6 to 1. So it's like, it just needs to pick up the net, one nearby, and that's fine.
Yep. And remember, it's not 6 to 1, it's 20 to 1.
Oh, excuse me. Yes, 21. Right.
Yeah, you got 6 million cones and 120 million rods. So even more than you thought, right? So thus, think about this. In complete darkness, there is no rod bipolar off cell that I know of that has been discovered, because the cone off machinery is already handling that process constitutively in complete darkness. But the moment, so you have this rod bipolar cell, and then I'm just saying it's on. But notice that the moment one small photon comes on, the cone is still sleeping and thus secreting glutamate, but the rods will pick it up. And of the hundreds of rods, one could have picked it up. That one little rod that picks up the photon, the one single photon, and transmits it, because 100 of them are connected to one rod bipolar cell. 100 of them send it to this one rod bipolar cell, who then says, let's hijack the machinery without waking up all of the cones, so you can actually see that there's some light, but you won't be able to make out color or anything. And this is an evolutionary trait we have. In the darkness, we can see, small photons of light, but we can't tell you if something's green, blue, or red. We can just tell you, hey, there's something here, I need to run, right? I mean, that's why we've developed it. Does this make sense why we would have this?
Yeah.
Okay. And then so I keep telling you it's an on because it makes sense, right? These guys are only active. I didn't tell, this is really only active. There's no rod off bipolar cell, because again, the cones machinery is kind of taking that because no ganglion cells are connecting to these rods specifically. So there's no need for an off or an on or anything like that. So they're using the rod bipolar cells only in the case of when there's light. So where would you expect that axon of the RB that you see here to be? Well, guess what? It's in the on layer, and that makes complete sense. Does that make sense?
Axons of the on layer.
Remember? So if it's an on bipolar cell, the axon of that bipolar cell is going to be in the distal IPL.
Yes.
That's what they mean by on layer, sublamina B.
Yep.
That's just another word that they use. The vision scientists call that the on layer. I call it sublamina B because that's actually the right thing, the way to say it. And then, but it's part of the IPL. And that's where you'd expect structurally, if I see a rod and I see something connecting to that rod that's going to the IPL, I sure as heck better see it go into the on layer or the distal IPL. Otherwise, I don't know what that what cell that is.
Yep.
Makes sense? Okay, so there you go. Okay, and then we're gonna get into some weird special ones. The S cones are weird. Just anything that connects to S cones have this odd tendency that breaks rules. Okay, so we're gonna look at it. One of them is this specifically to the S cone. And remember, S is blue, short wavelength. It's this blue bipolar cell or S cone bipolar cell. And then there's this less understood weird, and this is weird. It becomes more obvious in future dates. Here's a guy that breaks all the rules. He is a giant, and then here's the word bistratified type of bipolar cell, which means he is a bipolar cell. He's clearly diffuse. Take a look at how he's drawn. He has a bunch of these little connections. Notice how little the connection of the flat midget, the invaginating midget, right? But the diffuse have a ton of little dendritic connections to different cones. And, just giant bistratified GBB, he has different cones and, stuff he's connected to. But then guess what? He has both an on and an off. He's in both sublamina. Isn't that weird?
Yeah.
And so there's going to be a reason why he exists, but he's super weird. We think he's part of the S-cone activation pathway, which happened kind of different from the red, green, and the rod kind of evolutionary tree. So their apparatus to interpret it are a little different, but it'll make sense when we get to ganglion cell layers, kind of what the ganglion cells do in response to these sorts of bipolar cells. But just know that there is this bistratified type. It's not always just on and off, and this guy's an on-off. He's like both, okay? And it's like, okay, I don't know why that is, but now I know most of them on or off, and now you can tell, okay? So I'm just showing you this kind of possible S-cone bipolar cell a little bit more in closely, and then here's your S-cone bipolar, and then there's this weird S-cone off midget bipolar cell, and it's a little, it's kind of weird. I think that this is just another flat midget bipolar cell, and we don't know if there's a special on, there's a special off S-cone, or it's just part of the the stratification of the flat midget bipolar cell. But this is just a flat midget off bipolar cell that happens to deal with S cones in particular, because blue cones are special, okay? Just to give you the sense that you know that. So I always put an X through it, and I just say, just remember that it's a flat midget bipolar cell that happens to be off. Like you've already know it. If you saw it and you said flat midget bipolar cell, you'd know one cone, monogamous, one midget bipolar cell. I know what that is. Okay.
Well, I'd want to forget it because it doesn't connect to the, it's an off bipolar cell, but it goes to the distal layer because this is still.
No, it doesn't. This doesn't. This is the off. This guy goes to the on. Okay.
Oh, sorry. Okay. So that first line is still right. Okay. That is, yeah.
Remember what direction we are.
No, I was thinking, that first horizontal line is not the demarker between sublamina A and B.
Because you can see here, one thing I'll just show you because I think it's important. Sublamina A is actually busted up into 1 and 2, and then sublamina B is 3, 4, and 5. So we actually know these to the degree of dividing this whole thing up, right? Which is crazy. Right? We know it to that level. I think this is something like, if I'm remembering correctly, like 10 or 15 microns. And we know with alarming almost regularity, they're that spatially like delineated that they will frond themselves into that zone when they evolve. And that's so specific because it matters that much where these bipolar cells end, because it gives you a sense of the visual spatial kind of situation that you live in environment wise. So it's very important that it is not even off by 10 microns. You get what I'm saying? So in this case, so S1 and 2, he can be like either in this sublamina A1 or 2, it's made up of only 1 and 2, and then 3, 4, and 5 are on. This is off and this is on. This is distal and this is proximal. Make sense?
Yes.
Okay, so X that. So here's your bipolar cell. We know it goes from outer plexiform layer where it connects with the photoreceptors, and that's where the midget and diffuse kind of situation occurs. And then the inner plexiform layer, and that's where the on-off kind of situation occurs. Does that make sense?
Yes.
Right. And now, by the way, like when you look at outer and inner, it's kind of hard to mess those up per se when you start thinking about what's happening in them, right? Because you're like, oh yeah, like the on-off is going to happen in the inner plexiform layer because they need to be by ganglion cells and that amacrine cell situation, right? That's where that happens, right? It doesn't happen in the outer plexiform layer because that's where the midget and diffuse connections to the cones and rods are, right? So there you go. Okay, we're going to do a little review here, all right? So I'm just going to point at something. You're going to tell me, whether it's diffuse. I mean, it's easy, but it's always good to go through whether it's diffuse or not, that quality diffuse versus midget, flat versus invaginating, one cone versus many cones, and then, on or off, and then whether or not, it's a, S cone or, I don't know. I mean, you could, or a rod or something like that. Okay. So I'm going to point to that guy. What would you say this type of bipolar cell is?
Well, it looks like it's connecting to multiple photoreceptors. It's diffuse.
Yep.
And it looks like it is terminating in sublamina A. So that means it's an off, a bipolar off cell.
You got it. So it's a diffuse off bipolar cell. And then we always know, because we use the word off together, but if someone were to ask you, you're like, oh yes, I mean off center.
Off center.
Right. And then, and then it tells them, oh yeah, he knows that the whole annulus thing, right? Then you can work through that knowing that that's what this guy is. Many cones, like you said, remember, and it's cones, right? Mostly. I mean, there are some situations where it's like cones and maybe one round, but I have not seen that.
That's right. You said photoreceptors originally, but mostly cones, but yes.
And I would argue, just say cones. That's what I'd say, because remember, the rods have one bipolar cell, right? We already talked about this, right? So it's like, I haven't seen very much data. Now, again, we're not total vision scientists. Like, I'm not like, I might be in the 95th percentile of understanding this stuff, but like, I'm sure the 99.99% guy is going to be like, oh yes, but I know one situation in which a diffuse bipolar cell in a rabbit does this thing, and you're like, okay, and connects to rods and cones. You're like, okay, great. I'm happy for you. But for our purposes, it's going to be easy to understand. Okay. So I'm going to point to him. What is this one?
So it's a midget off center bipolar cell.
What other quality can you say that is?
Are we looking?
What is this? What does this mean? What's that letter mean?
Oh, that's flat.
Good, flat. So it's on the surface of the cone, right? Just so you know, pathologically, if someone, by the way, this is in your book, right? They show you that like schema. So I just wanted to know, like, yeah, this one's going to be flat, midget, bipolar cell. And you said off or on. Which one did you say?
Off.
Off. And thus, how many cones? One or many?
Because it's midget, it's just one.
Right, monogamous, right? So flat, midget, off, bipolar cell, one cone. You got it, okay? How about this guy?
It's a diffuse, so I can't tell if it's going into, it's kind of, I'd say it's off.
Yeah, good. I'd say that too.
So diffuse off bipolar cell.
Good. You don't know flat or
cones.
you got it. So diffuse off bipolar cell. Perfect. And you said many cones. How about this one?
And it's a invaginating midget bipolar on cell.
You got it. Invaginating midget bipolar on cell, one cone, right? Because it's a midget.
Yeah.
Now I want to just point something out. It just so happens why we talk about flat and invaginating, and the reason why they specify it is because when you see a flat midget, like that's the only cone it's with, and then you see an invaginating midget, that's the only cone it's with, just by virtue of seeing invaginating versus flat, you don't actually necessarily in a primate retina have to go and look at the IPL. You know the invaginating is going to be an on and the flat's going to be an off. It just happens to be concordant. It's not true in every species, I believe, but in ours, it is. And so if you ever are like looking and you're pathologic, you're going to impress a lot of people. You'll be like, yeah, but that's at the center of the cone. And I know that's the only connection that bipolar cell has to that, that's the only cone it connects to. And it's on the surface and it's a little, it's a midget connection. It's not diffuse. Okay, I'm going to guess that that one's going to be an off. It's going to terminate in sublamina A. I don't even need to look, because it's a primate retina. It's a human. You get what I'm saying?
Yeah.
So it's kind of cool. I don't know. It's like putting together a different way of determining the function of something just by its structure, because that is very much retina, right? I mean, retina is so literally like where you are matters, right? In terms of what information you're giving. It's like, I guess it's the literal classic organ of the body that does this, right? Because that's its job. So why wouldn't these guys be like that? What you see in their connectivity is going to give you a sense of their physiology, right? And so I think that's a great thing to remember. Okay. All right. So how about this guy? What is his situation?
So it's diffuse on bipolar cell.
And
so
what
many cones.
Well, take a look. I pointed to him, and then it has this like letter next to it.
Oh, so it's a rod bipolar cell.
So it's a rod bipolar cell, and so the rod bipolar cell, does he connect to a ganglion cell or no?
No, he talks to the amacrine cells.
He talks to the amacrine cell. There's a special amacrine cell, by the way. It's not even cells. There's like one cell that's super famous in the, famous within the vision scientist world, but it's super famous for this particular function. Okay. And then remember, when we talk about diffuse, we're talking about this side of him, remember? Because that's the diffuse side. Here we go. These processes, they're, we haven't talked about that, right? But we know that he's in the on layer, right? Because the rod bipolar cell is on, right? That's what he is. I mean, it just happens to be, that's his function. Okay.
Yes.
All right. So we said many rods. Okay. And then tell me, like, what is this one? Tell me.
Is this the S cone?
Yeah, you got it. Yeah. So what is it though? Is it diffuse, do you think, looking at this like schema?
Looking at it, looks more diffuse than the midgets that we've seen. So I'd say diffuse, yeah.
You'd say diffuse, and then you'd say on or off.
Say on.
You'd say on, right? Again, distal IPL, right? You'd say on. And so, and then if I look over here, what is this guy?
That is also diffuse. And this is the one that's going to both.
Yeah. What do we call it?
The great.
Yeah, the giant bistratified bipolar cell. And you were thinking it, these massive ones, when you do pathology, you can see these massive cells that are in this layer from the internuclear layer, and that's coming out, towards the IPL. And you're like, whoa. And we saw these and it was like, wow, what is this? And they've got on and off. These are two strati, I guess. So stratuses, right? So it's like, you've got a bistratification here, right? That's what they're calling. So it's a giant bistratified bipolar cell. It tends to deal with the blues and the kind of the weird stuff, and we'll get more into that on ganglion cells. So like you said, here was the on, and then here's many cones, as you said, because it was diffuse, and then your giant bistratified bipolar cell, which also probably is, we don't know, but probably diffuse, and we can kind of look at it again, structure and function. And then, and then that's that. I just wanted to show you again, the S cone off, right, to turn off is actually almost like a flat midget cell. So it's like, your S cone on is weird, right? It's like a bipolar cell, and then your S cone off is a midget. I don't know. But the point being is that they're special, but you got it. Okay. So that's that for this session. Okay. Next time we're going to do ganglion cells. Okay.
Great.
Yep. Helpful, I hope.
I love it. Yes. Thank you so much.
Okay, no problem. And I'll send along the handout. Okay. And get some rest, my friend. Okay.
I will do. Thank you so much, Dr. Diaz.
Okay, no problem. Okay, bye.